Welcome everyone to this talk from my room, which was originally supposed to happen in Los Angeles at the American Association of Physical Anthropologists annual meeting in the session Forensic Anthropology, Methods and Approaches. My name is Emese Vegh. I'm a third-year PhD student at the University of Oxford. The other authors on this paper are Andrea Czermak, Nicholas Marquez-grant and Rick Schulting. Our title is Detection of Early Postmortem Changes in Burnt Bones Through Histotaphonomical Analysis. So here is the overview of the talk. I will give a bit of a background on bone diagenesis and burning of bones, then move on to the research question and rationale, experimental outline, methods, results and discussion, and then we are going to conclude. As we all know, bone is a complex composite material. On the left, you can see a cross-section of this cortical bone from a long bone. In this study, we are focusing on the microstructure of cortical bone. So bone undergoes diagenetic alterations post-deposition. Three distinct pathways can be distinguished, and we are focusing on microbial degradation of both the inorganic and the organic phases. This degradation presumably happens quickly after death, as it is thought to be linked to putrefaction processes, while soft tissues are still present. Many studies use histotaphonomic features on unburned bones to inform on the initial postmortem period of the body. These are microscopic focal destructions, or MFDs. These can be linear, budded, or lamellate, and these are thought to be caused by either endogenous bacteria from the gut or bacteria from the soil. And then there are Weddell tunnelings, which are thought to be caused by fungi as an indication of surface exposed or defleshed buried bones. And then there are Weddell type 2, which are these little spider-like structures. This happens when the lacunae and the canaliculi are both enlarged, and these are thought to be caused by fungi or bacteria, depending on which paper you read. And then there are cyanobacterial tunnelings, which are caused by cyanobacteria from marine or freshwater based context. So these features are usually named after they're thought to be causative agents. Because of this, there is a disagreement and a confusion in the literature about these features, and often the same features are called differently and thus interpreted differently. And here we are using the terminology and the physical appearance of the features written down by Brönnimann et al. 2018. So let's move on to burning. The purpose of burning can be the destruction of the body, homicide or suicide. There can be accidental fires as well, which result in death of the victim, such as wildfires and building fires. The two most important factors are the heating temperature and the duration of the heat. Bone goes through a series of changes through burning, such as color change or different heat-induced fracturing. Burning of the body can happen either when the body is fully flashed or once it has decomposed to different degrees. For example, when the burning happens to conceal evidence of murder at a later date. Early studies mainly focused on establishing whether the bones were at either of the two extremes of the spectrum when burned, so whether they were fresh or dry. These were primarily macroscopic techniques based on the presence of thumbnail fractures and warping, but these were shown to be unreliable. There are only relatively a few studies about burn bone histology, which mostly focus on what the fire temperature and duration was, what the species of the burnt tissue was, and establishing age at death from burnt bones. There is a disagreement in the literature at what temperature structural changes first occur in bone. It was reported in different studies that the bone's histological structures is nearly identical to unburned bones when they are burnt under 600, 700, 900 or 1200 degrees. It is also not clear whether these temperatures refer to bone temperature or the fire's temperature. Additionally, the discrepancy might be due to the different methods used for burning. So for example, burnt in a furnace or in an outdoor fire. The research question of this study was whether we can learn if bones were fresh or decomposed when burned from microbial bioerosion signatures. Identifying bioerosion on burned bones would mean that it may be possible to establish whether the bone has gone through a phase of decomposition when the soft tissue was still present prior to burning. This research therefore has implications to understanding the sequence of events leading to deposition in forensic investigations, as well as in funerary practices in the archaeological record. There are a few cases discussed in the forensic anthropological literature in which a single individual has been found partially burnt. In these cases, the postmortem intervals were established using both the sign of gnawing and the pattern thermodestruction. However, both burnt and unburned from the same individual are rarely available for analysis, and all we are left with is fragmented burnt remains. There is a need for better and more accurate methods from a single burnt skeletal fragment. Experimental outline. First, flashed pig TBA were placed in a cage on a field at Wytham Woods, Oxfordshire, England. This was done in several rounds, and then the legs were left to decay, collected, and then subsequently burnt after 2 weeks, 1 month, 3 months, 6 months, and 1 year. Additionally, fresh fleshed legs were served as unburnt and burnt control samples, so they were burnt as well. The bones were burnt until they turned mostly white or calcined. After this, two methods used were optical transmission microscopy and backscattered electron microscopy, or BSEM, on the electron microprobe. First, the unburned bones were degreased in a mixture of methanol and chloroform for up to 20 weeks, and then the unburned and the burnt bones were embedded into epoxy resin, and then a Buhler saw with a diamond blade was used to cut out 50 to 70 micron thin sections out of the samples, and then these were fixed onto glass slides, and pictures were taken under the transmission light microscope at varying magnifications, and then a data labeling application was built using Python and JavaScript programming languages. So to be able to take compositional images on the BSEM after the bones have been embedded into resin, they were ground with carbon papers of progressively finer grit size and then polished with diamond paste. Then the samples were carbon coated in order for the carbon to act as a conductive layer to improve signals and hence the image. This was done because it has been suggested that actual microbial bioerosion features can only be revealed by demineralized or dark foci with hypermineralized or white rims around them. There is an example here from Turner Walker 2019 who showed that although features might appear as tunneling under the optical microscopy under the BSEM, they do not show the difference in compositional difference, as you can see on the most lower right image. So this is how the data labeling application looks like. Only the 50 times magnification images were used, and 3 to 5 of them per sample. Each image was divided into 100 equal blocks. The random selection of images by the application excluded bias in labeling. When a feature was present, I clicked on the feature and then I click Save. The results are automatically saved in a database created by the application, and the cumulative score for the 100 blocks presents a percentage score for each image. And I labeled 27,000 of these blocks. And of course, as we've seen before, the features were labeled based on the image on the lower left taken by Brunimon et al. 2018. There were some problems I came across when labeling. For example, these tunnels are obviously different, but based on the criteria by Brunimon et al. 2018, I labeled them all as cyanobacterial tunneling. Some of these are probably just Volkmann's canals, for example, the two images on the left. So let's look at some of the selected histological images from different postmortem intervals of the unburnt and burnt bones. The first one on the left is an unburnt fresh control bone, and right next to it, two fresh burnt bones. As you can see, Weddell too, so the little features that resemble spiders. that is present on the freshly burned bones, and they appear on the unburned bones after just 2 weeks of decomposition. This was also the second most common taphonomic feature following hairline cracks. The fresh unburned bones had almost no features identified as Weddell tunneling, Weddell 2, lamellate, or budded MFDs, and showed a minimum amount of cyanobacterial tunneling, but it can't be cyanobacterial tunneling, so these are almost certainly mislabeled Falkman's canals. On the right, there are two images showing examples of budded MFD and lamellate MFD. The first image is on a 6 months postmortem unburned section, and the second one is on a 1-year-old postmortem burn sample. The graph on the left shows the percentage of given features present for each decompositional stage before and after burning. On the x-axis, you can see the group starting from the control samples or 0 days postmortem to 1 year postmortem. Weddell 2, what we were looking at the slide before, are assigned to bars with blue colors. The table on the right shows the results of a linear regression, where the null hypothesis was that the presence of X feature per bone does not increase with a postmortem time period. As you can see in the unburned groups, Weddell tunneling, lamellate MFD, budded MFD, and hairline crack showed statistically significant p-values, while groups that were burned only showed Weddell 2 and budded MFD as significant. We also calculated the coefficient of variation for each feature, which showed that the greatest variability was present for the lamellate and the budded MFD features, meaning that these features were the least reliably present across all images. So let's move on to the BSEM results. The first image here is from a 1 month unburned bone. The second is from a 3 month unburned bone, and the third is from a 6 months burn sample. The BSEM results show that there is some dark demineralized areas and adjacent hypermineralized areas present, for example, on the first image around cracks or tunnels. But on the second image, we can see these around enlarged lacunae and canaliculi, but these look nothing like bioerosion in Turner Walker's paper from 2019, and all these compositional differences are obliterated by burning, as you can see on the image on the right. The contrast difference here that you can see is due to the burning process. 3 features associated in the literature with microbial bioerosion were present on the burned fresh control group, but absent on the fresh unburned group. Weddell 2 was a common feature on both the unburnt and the burnt bones after only 2 weeks, and we suggest that these are trapped carbonized organic material in the lacunae in the fresh burnt bones. The presence of Weddell tunneling, budded, and lamellate MFD had a statistically significant relationship with time since deposition, but only budded MFD was still significant after burning. However, this significance was less robust after burning, and this feature also had a very high coefficient of variation. And this suggests that budded MFD might have the potential to be an indication of bioerosion, but further research is needed. Additionally, this feature appeared on the unburned bones after just 1 month, while the literature says that this first appears after 3 months. However, on burnt bones, we were only able to see these after 3 months. And then cracking and hairline cracks were more associated with burning than with decay. Cracking showed a relationship with time since deposition on the burnt bones, but both of these features were produced both by laboratory preparation of cutting and fixing onto glass slides. So these are not reliable features of decay. To conclude, our study has been the first histotaphonomic research to apply quantitative analysis to calculate the percentage of areas affected by bioerosion in unburnt and burnt bones. Our results showed that most of the thought to be microbial bioerosion features on both unburnt and burnt bones are not reliable indicators of putrefaction or decomposition, because these appear on the freshly burned bones as well. Thus, these can be caused by other factors, as we've seen, such as burning. Budded MFD was the only feature that might be used as a proxy for decomposition, but its visibility was reduced by burning. Cyanobacteria tunneling are probably Folkman's canals, and we have shown that they do survive intense burning. And finally, although BSEM is a useful technique in microbial bioerosion studies on unburnt bones, it cannot be used in burnt bones. So to conclude, microbial bioerosion features are not accurate proxies for the body's pre-burning condition, and caution should be practiced when identifying these in both forensic and archaeological cases. Thank you so much for the attention.